Basics of Medical Biochemistry. First edition, 2023. Chapter 19. Molecular Genetics, Part 2 of 2. By Professor Dr. Muhammad Jawad. MBBS, Dao, M.Phil Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College, Air University-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. 19.3 Recombinant DNA Technology. Recombinant DNA, rDNA, is a form of DNA that does not exist naturally. It is created by artificial means when a gene of one species is transferred to another living organism. This is known as recombinant DNA technology or genetic engineering. Recombinant DNA technology was developed by two scientists named Boyer and Cohen in 1973. They produced the first plasmid vector capable of being replicated within a bacterial host. Recombinant protein is derived from recombinant DNA. Recombinant DNA is produced through the addition of relevant DNA into an existing organismal genome, such as the plasmid of bacteria, to code for or alter different traits for a specific purpose, such as immunity. It differs from genetic recombination, in that it does not occur through natural processes within the cell or ribosome, rather it is exclusively engineered. 19.3.1 Goals of recombinant DNA technology. To isolate and characterize a gene. To make desired alterations in one or more isolated genes. To return altered genes to living cells. To get new genes through artificial synthesis. To do the required alteration in the genome of an organism. To understand hereditary diseases and their cure. To improve the human genome. 19.3.2 Steps of recombinant DNA technology. 1. Procedure of making rDNA. It includes the following steps. I. Extraction of DNA. Cells are lysed with detergent that disrupts the cell membrane. Cell contents are treated with protease and RNAs to destroy the protein and RNA respectively. 2. Cutting of DNA. DNA can be cut into small fragments by mechanical shearing. Restriction endonucleases are used for this purpose. They act as scissors of molecular genetics. The cut DNA piece has two blunt or sticky ends. Restriction enzyme. It is a special class of sequence-specific enzymes, found in bacteria. It is site-specific, i.e., can cleave DNA molecules only at specific nucleotide sequences. Restriction enzymes can recognize DNA-based sequences that are palindrome. Restriction enzymes make staggered cuts with complementary base sequences for easy cyclization. Plasmid is also cut by this enzyme to make a gap where foreign DNA is inserted. Table 19.1 Enzymes used in recombinant DNA technology. Name of enzyme. Function. DNA ligase. Binding of DNA molecules. Type 2 restriction endonuclease. Cleaves DNA at specific sites. Reverse transcriptase. Synthesize a DNA copy of an RNA molecule. DNA polymerase I. Fill single-stranded gaps of DNA duplex. Polynucleotide kinase. Transfers terminal phosphate, gamma position, from ADP to 5O groups of DNA or RNA. Terminal transferase. Adds homopolymer tails to the 3O ends. Exonuclease 3. Removes nucleotide residues from the 3 ends. Bacteriophage, lambda, exonuclease. Removes nucleotides from the 5 ends. Alkaline phosphatase. Dephosphorylates 5 ends of RNA and DNA. 3. Isolation of DNA. The DNA piece of the desired length is separated from the cell debris. 4. Joining of DNA fragment to vector. The joining of DNA fragment to a suitable foreign molecule of carrier DNA is carried out by the enzyme DNA ligase. As a result of this union, recombinant DNA molecule is produced. It is obtained from two sources. Vector. it is a DNA molecule into which a gene of interest, cut DNA fragment, is inserted to get an rDNA. Vector is used for cloning as it is capable of replication in the host organism. The vector may be a plasmid, a bacteriophage, bacterial artificial chromosomes, or yeast artificial chromosomes. Plasmids are small, extrachromosomal circular DNA molecules found in certain bacteria. Plasmids are also found in some strains of yeast. 2. Insertion of rDNA into the host cell. The rDNA is inserted into the host cell, which is usually a bacteria, prokaryote, example E. coli, or it may be yeast cells, for example eukaryotes. The host cells are treated with calcium chloride to make them more permeable for the take-up of rDNA. 3. Multiplication of host cell with rDNA. When the host cell divides and redivides, the replication of rDNA also occurs simultaneously. The copies of rDNA molecules are passed to offspring and further division of vector takes place. Thus, the DNA is amplified. As a result of repeated cell division, a colony or clone of host cells containing copies of rDNA molecules is produced. 19.3.3 Hybrid DNA or chimeric DNA. The type of rDNA obtained by joining the human gene with the non-human cloning vector is called hybrid DNA or chimeric DNA. The rDNA can be further modified according to the required need. 19.3.4 Applications of rDNA technology. 1. Medicine, gene therapy, antiviral therapy, vaccination, synthesizing clotting factors and lung surfactant. 2. Pharmacology, artificial insulin and other hormone production, drug delivery to target sites, etc. 3. Agriculture, growing healthy crops of choice in large quantities, pesticide-resistant crops, and fruits with attractive colors, all being grown in artificial conditions. 4. Other uses, mass production of proteins, fluorescent fishes, production of oil-eating bacteria which are used to clean up the oil spills at beaches. Figure 19.11 Steps of recombinant DNA technology. 19.4 Genetic testing. Genetic testing is a type of medical test that identifies changes in chromosomes, genes, or proteins. It can also be used to determine a child's parentage. Genetic testing in a broader sense includes biochemical tests for the possible presence of genetic diseases or mutant forms of genes associated with an increased risk of developing genetic disorders. Genes are the working subunits of DNA. DNA carries the complete set of instructions for making all the proteins a cell ever needs. Each gene contains a particular set of instructions, usually coding for a particular protein. Some genes play a role in the early development of the embryo and are then shut down forever. Some genes enable cells to make proteins needed for basic functions. Gene mutations can be either inherited from a parent or acquired. A hereditary mutation is a mistake that occurs in the DNA of virtually all body cells. Hereditary mutations are also called germline mutations because the gene change exists in the reproductive cells and can be passed from one generation to another generation and from parent to newborn. Extensive research in the recent past has devised many new techniques which allowed scientists to learn a great deal about how genes work and how genes are linked to disease. Now available methods can identify mutations and changes within genes that can lead to specific disorders. Tests for gene mutations make it possible not only to detect diseases already in progress but also, in certain situations, to foresee diseases yet to come. 19.4.1 Purpose of doing genetic testing. These include finding genetic diseases before the birth of babies, finding out whether people carry a gene for a disease and the possibility of its passage into their children, screening of embryos to detect any disease, testing for genetic diseases in adults before the appearance of symptoms, confirming a diagnosis in a person who has disease symptoms, suggesting the type and dose of a medicine that is best for the management of a certain disease. 19.4.2 Methods of genetic testing. Several methods can be used for genetic testing, such as molecular genetic tests, or gene tests, study single genes or short lengths of DNA to identify variations or mutations that lead to a genetic disorder. Chromosomal genetic tests analyze whole chromosomes or long lengths of DNA to see various genetic changes, such as an extra copy of a chromosome, that cause a genetic condition. Biochemical genetic tests study the amount or activity level of proteins. Abnormalities in their quantity or performance can indicate changes to the DNA that result in a genetic disorder. 19.4.3 Types of genetic testing. Following are the important types of genetic testing. Diagnostic testing. It is used to diagnose or rule out a specific genetic or chromosomal condition. This type of test commonly detects a specific gene alteration but is often not able to determine disease severity or the age of onset. It is estimated that there are greater than 4,000 diseases caused by a mutation in a single gene. Newborn screening. It is used just after birth to detect genetic disorders that can be treated early in life to avoid a fatal outcome. Carrier testing. It is used to identify people who carry one copy of a gene mutation that, when present in two copies, causes a genetic disorder. Pre-implantation genetic diagnosis. Genetic testing procedures that are performed on human embryos before the implantation as part of an in vitro fertilization procedure. Prenatal diagnosis. It is used to detect changes in the genes or chromosomes of the fetus before birth. This type of testing is offered to couples with an increased risk of having a baby with a genetic or chromosomal disorder. Predictive and pre-symptomatic testing. These types of testing are used to detect gene mutations associated with disorders that appear after birth, often later in life. These tests can be helpful to people who have a family member with a genetic disorder, but who have no features of the disorder themselves at the time of testing. Pharmacogenomics. It is the type of genetic testing that determines the influence of genetic variation on drug response. Forensic testing. Forensic testing uses DNA sequences to identify an individual for legal purposes. This type of testing can identify crime or catastrophe victims, rule out or implicate a crime suspect or establish biological relationships between people, example paternity. Parental testing. This type of genetic test uses special DNA markers to identify the same or similar inheritance patterns between related individuals. Research testing. Research testing includes finding unknown genes, learning how genes work and advancing further understanding of genetic conditions. 19.4.4 Genetic counselors. These are health professionals having specialized education, training, and experience in the areas of medical genetics and counseling. They are an integral part of the healthcare team providing information and support to individuals and families who have members with birth defects, or genetic disorders or may be at risk for a variety of inherited disorders. Genetic counselors also serve as educators and a resource for other healthcare professionals and the general public. 19.5 Polymerase chain reaction, PCR. It is a technique widely used to rapidly produce millions to billions of copies of a specific DNA sample. PCR is often considered one of the most important scientific advances in the field of molecular biology. It is very advantageous to take a very small sample of DNA and amplify it to a large enough amount to study in detail. To amplify a segment of DNA using PCR, the sample is first heated so the DNA denatures, or separates into two pieces of single-stranded DNA. This process results in the duplication of the original DNA, with each of the new molecules containing one old and one new strand of DNA. 19.5.1 Requirements for PCR. 1 DNA template. A DNA template is the DNA molecule that contains the DNA segment to be amplified. The concerned segment possesses the target sequence. 2 DNA polymerase. It sequentially adds nucleotides complementary to the template strand at 3' of the bound primers and synthesizes new strands of DNA complementary to the target sequence. The most commonly used DNA polymerase is TAC DNA polymerase derived from Thermus aquaticus, a thermophilic bacterium having high temperature stability. 3 primers. These are synthetic DNA strands of about 18 to 25 nucleotides complementary to the 3 ends of the template strand. DNA polymerase starts synthesizing new DNA from the 3 ends of the prime. Primers should bind to the template with good specificity and strength. If primers do not bind to the correct template, the wrong sequence will get amplified. Optima primer sequences and appropriate primer concentrations are essential for maximal accuracy in PCR. 4. Magnesium ions, Mg2+ ions in the buffer act as a cofactor for the DNA polymerase enzyme. Magnesium affects primer annealing and template denaturation, as well as enzyme activity. 5. Deoxyribonucleotides, all 4 deoxyribonucleotides, DATP, DGTP, DCTP, DTTP, are required. 19.5.2 Procedure of PCR. Following are the major steps in a PCR, which are repeated for 30 or 40 cycles. This is done on an automated cycler, which can heat and cool the tubes with the reaction mixture in a very short time. 1. Selection of DNA segment. The specific DNA segment is selected, which acts as a template for the synthesis of its copies in multiple numbers. 2. Denaturation. It is carried out by heating up to 94 degrees Celsius for 1 minute, which causes the separation of DNA double-stranded. Now, each strand acts as a template for the synthesis of a complementary strand. 3. Annealing at 54 degrees Celsius. This step consists of cooling of reaction mixture down to 54 degrees Celsius, which causes hybridization, annealing, of primers to separate strands of DNA, template. The length and guanine cytosine content, GC content, of the primer should be sufficient for stable binding with the template. Higher GC content results in stronger binding. If GC content is less, the length may be increased to have stronger binding. 4. Extension at 72 degrees Celsius. The reaction mixture is heated to 72 degrees Celsius, which is the ideal working temperature for the TEC polymerase. The polymerase adds nucleotide complementary to the template on 3O of primers, thereby extending the new strand. 5. Control step. The steps of denaturation, annealing, and extension are repeated 35 to 40 times to produce millions of exact copies of the target DNA. Once several cycles are completed, during the control, hold, step, a 4 to 15 degrees Celsius temperature is maintained for short-term storage of the amplified DNA sample. As both strands are copied during PCR, there is an exponential increase in the number of copies of the gene. Suppose there is only one copy of the desired gene before the PCR starts. After one cycle of PCR, there will be 2 copies, after 2 cycles of PCR, there will be 4 copies, after 3 cycles there will be 8 copies, and so on. 19.5.3 Applications of PCR. This technique is used extensively in molecular biology and genetic disease research to identify new genes. For example, the sample containing pathogenic DNA can be PCR amplified using different known specific primers. The amplification indicates the presence of pathogenic DNA. In the fields of anthropology and evolution, sequences of degraded ancient DNAs can be tracked after PCR amplification. PCR has been used for wartime human identification and validated in crime labs for mixed sample forensic case work. PCR permits early diagnosis of malignant diseases such as leukemia and lymphomas. PCR assays can be performed directly on genomic DNA samples to detect specific malignant cells, and infectious agents like mycobacterium, anaerobic bacteria, or viruses. This technique is also used for the introduction of mutations at the desired place in a DNA sequence. Summary. Molecular genetics is the study of the heritability of traits at a cellular level. It is more concerned with the studies on DNA and the cellular mechanisms of trait transmission between cells. Humans have 22 pairs of autosomes and 1 pair of sex chromosomes, which are X and Y. Sex chromosomes help in determining the sex or gender of an individual. Females have 2 X chromosomes in their cells, while males have both X and Y chromosomes in their cells. Complementary DNA, cDNA, is synthesized in the laboratory from messenger RNA. Recombinant DNA, rDNA, molecules are DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning to bring together genetic material from multiple sources. The coding part of a gene is known as an exon, whereas the non-coding region is termed the intron. Codon is a sequence of 3 successive nitrogenous bases in the molecule of mRNA. Penetrance refers to the probability of a gene or trait being expressed. One example of this is polydactyly in humans, i.e., extra fingers and or toes. About 30% of DNA is composed of genes, but only 1.5% is involved in coding, while the remaining 28.5% acts as non-coding. Humans have approximately 30,000 to 35,000 genes arranged on their chromosomes. An allele is an alternative form of a gene, one member of a pair, that is located at a specific position on a specific chromosome. If the alleles of a gene are different, one allele will be expressed. It is termed the dominant gene. The effect of the other allele is masked and it is called a recessive gene. A phenotype is an individual's observable traits, such as height, eye color, and blood type. Heterozygous refers to having inherited different forms of a particular gene from each parent. Homozygous is a genetic condition where an individual inherits the same alleles for a particular gene from both parents. Codominance is a relationship between two versions of a gene. In codominance, neither allele is recessive nor dominant, rather the phenotypes of both alleles are expressed. Gene expression is the process by which the information encoded in a gene is used to direct the assembly of a protein molecule. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. DNA sequencing is a laboratory technique used to determine the exact sequence of bases, A, C, G, and T, in a DNA molecule. The type of rDNA obtained by joining the human gene with the non-human cloning vector is called hybrid DNA or chimeric DNA. Cloning is the process of making identical copies of an organism, cell, or DNA sequence. Epigenetic changes alter the physical structure of DNA. By the technique of genetic engineering or recombinant DNA technology, a large variety of useful substances like hormones, example insulin, antigens, antibodies, and many other therapeutic agents have been produced. Polymerase chain reaction, PCR, is a technique widely used to rapidly produce millions to billions of copies of a specific DNA sample. The technique of PCR is used extensively in molecular biology and genetic disease research to identify new genes. References. Exercise questions. MCQ 1. Which of the following techniques is used to identify all of the genes that are active in any given cell under any given set of conditions? cDNA cloning, genomic cloning, northern blotting, polymerase chain reaction, PCR, southern blotting. MCQ 2. A 6-year-old girl suffering from recurrent vasculo-occlusive episodes presented with complaints of severe bone pain often. It is suspected that she is suffering from sickle cell anemia. Which of the following tests would be appropriate to confirm this diagnosis? cDNA cloning, genomic cloning, polymerase chain reaction, restriction fragment length polymorphism, southern blotting. MCQ 3. Which of the following enzymes is required to generate a genomic library? DNA polymerase, DNase I, polynucleotide kinase, restriction endonuclease, reverse transcriptase. MCQ 4. A group of geneticists is carrying out experiments designed to utilize DNA encoding a wild-type gene to treat a disease resulting from the inheritance of a defective gene. Which of the following techniques would be the most appropriate to achieve the purpose of these experiments? Gel electrophoresis, gene therapy, polymerase chain reaction, RNAi, transgenesis. MCQ 5. A study is designed to analyze the pattern of genes expressed in breast cancer to identify differences in normal breast tissue. Which of the following enzymes is most useful in this study? DNA ligase, DNase I, polynucleotide kinase, reverse transcriptase, restriction endonucleases. End of Part 2 of 2 of Chapter 19. Thank you.